Hey guys, so we've just gone over EcoBAM transfers and assemblies, and now we're gonna look at assembly a little bit more. So we just assembled a left part and a right part, and now we're gonna start to look at assembly trees. But before we go down that, I just wanted to clarify one thing I think I may have glossed over. So the difference between an EcoBAM transfer and an assembly. So an assembly is you have A, B, C, D, and an assembly is you put A and B together, you put C and D together, then you get A, B, and C, D. And then you put A, B together and C, D together to get A, B, C, D. That's an assembly. An EcoBAM transfer is you have A somewhere. It could be in a plasmid, it could be on the genome, it could be on some PCR fragment. I don't know how to represent that in a way that looks different from the genome, but it could be anywhere. And the EcoBAM transfer is you take that and then you put it into a standard biobricking plasmid. So you have your EcoBagel-BAM zoocyte. And that usually involves PCRing it off of the genome or whatever it is, then digesting it, and then transferring it into the plasmid. However, an EcoBAM transfer can also mean transferring a part from one plasmid, we'll say plasmid F9 to plasmid G6, and we would just, digest. Here, let me change colors, too much red here. We would we would digest eco, digest BAM, same thing here. We would digest eco, digest BAM, and we just move on over. That would ligate into there, that would ligate into there, and we get EcoBAM transfer from one plasmid to another. So in order to do an assembly, if you wanted to assemble A and B together, A and B need to be in the same plasmids. So if we're using these codes or these little names that I just made up, they don't really mean anything, we would have plasmid G6 and G6. And they would have to be in plasmid G6 and G6. If we had plasmid G6 and A3, then the zoocyte could maybe go through some sort of antibiotic resistance, or maybe we're not using the zoocyte, maybe we're using some other enzyme, and it could be cutting the ORI in half. So you can be cutting up important parts of your plasmid. So that's why it's easier just to put all of your parts into one vector backbone. Well, it's not easy, it's necessary if you want to do this assembly tree. And then from there you can actually do the assembly. So we got a close-up look at an EcoBAM transfer and one assembly step. So now I'm just gonna want to zoom out and take a little bit of a higher elevation look. So if we go back to our favorite letters, A, B, C, and D, and these are all parts, each of which is in its own vector backbone. And they all have their EcoBagel-BAM zo, EcoBagel-BAM zo, EcoBagel-BAM zo, EcoBagel-BAM zo, along with their primer sites, which we won't put in for now. You can see that the assembly is pretty easy. So in order to put these together, we would digest there and there, so that would be a BAM zo, and we'd do a bagel zo, and those two would come together. We just ligate, and let me go back to my green. So this would be a round one assembly, and you'd get A with your scar and your B. EcoBagel-BAM zo. So we reconstituted that zoocyte during that ligation, and we made a scar site up here. And just to be extra clear, we're getting rid of these inner things. So that's all gone. And we do the same thing here. We would cut at that BAM-ZO for that left-hand part, so we'd cut right there. And we would cut at bagel-ZO for that right-hand part. And again, the reason we're using bagel and BAM is because they have compatible sticky ends. And once we ligate these together, we get a composite part. So we're going to have C, D, and we're going to have Eco, Bagel, BAM, ZO with our scar sequence in between. We'll just put an S there for our scar sequence. So that's the first round assembly. So you have your basic parts are going to be here. Oops, can't spell. Basic parts. Then you have your round 1 assembly. Then you just repeat the process. So you would digest BAM-ZO, and you'd cut right there, get rid of that chunk, and you get rid of it by running it on a gel and cutting out the fragment you want. And then here we're cutting bagel-ZO. And then we can ligate them together. See if I can do this all on one page. So we have A, then we have our first scar sequence, and we have B, then we have a scar sequence. And that's the scar sequence we just made, so we'll put a star by it. That's the most recent scar sequence. Then we have C, then we have an old scar sequence, then we have D, and then the rest of our plasmid. Eco, Bagel, BAM, ZO. And that would be an example of an assembly. So let's talk about a few contingencies. Every now and again, it actually happens more than I'd like to admit. Let's say you want to do your BAM-ZO, Bagel-ZO digest. And in this one, your BAM-ZO, this small fragment is 1 kb, and this fragment is 3 kb. So that's cool. But then you do your Bagel-ZO digest here, and then this fragment that you want to cut out is 2 kb, but then this fragment is also 2 kb. That's a problem. When you run it on a gel, you need to be able to resolve your two bands and cut out the one that you want. But if they're both 2 kb, you're going to have one band here, and then you're going to have another band right on top of it. And you're not going to be able to cut them out. And when you try to do that assembly, you're just going to get a bunch of background. Instead of getting A, B, C, D, you're just going to get a bunch of these plasmids religating together, and it really sucks. So what you do instead, in that case, is you look for other restriction sites. So if you can't use the ZO here let's change colors to, how about, we haven't used pink in a while. You're going to have to look for something else other than ZO. So you're pretty much stuck with BAM and Bagel. Those are good. That's the only way you're going to be able to make that junction. But you have flexibility when it comes to the other one. So you want to find an enzyme that's maybe somewhere here, or I don't know, anywhere, and then you want to find an enzyme that cuts well and will give you different fragment sizes. So maybe this one will be 2.5 and then 1.5, and this one will give you, who knows, 1.5 and then 2.5 over here. So Some enzymes that I commonly use would be ALWN1. I think that's what it's called. Maybe it's ALNAWNL1. I can't remember, but it's something like that. AVR1 is another one that I use. But if you're just using APE or something like that, you can just press Control E. Or if you're going to be using Clotho, there's also an enzyme function that you can display all the unique enzymes for. And you're going to be looking for the unique enzymes in that backbone. So just find a good enzyme and substitute that for zo. So instead of doing bagelzo bamzo, you're going to be doing bagel whatever your enzyme is, and then a bam whatever your enzyme is. And the point is that you really need to make sure that you're getting resolvable bands that you can cut out and then proceed with your assembly. Let's see if I can think of anything else important for assembly. So another important thing is to always pay attention to your backbone. So I was mentioning that we have antibiotics and origins of replication. So we'll just draw these into our backbone. The antibiotics, everybody's trained from a very young age. Well, maybe not everybody, but you're trained from when you start doing your research in a microbial lab to pay attention to your antibiotic resistance. That's kind of second nature to everybody. So we'll put resistance. So just do that. That's, common knowledge. But here, our origins of replication we don't always think about those so much. So we have some origins of replication that will work in any bacteria, and the origin of replication is exactly what it sounds like. It's a piece of DNA from which the plasmid replicates itself. And it also determines the copy number. So a strong origin of replication will give you like 500 plasmids, maybe 800, who knows. I think like Puck can be that depending on the context. And a weaker origin can be 50 copies of the plasmid per cell. Some origins are temperature sensitive, meaning that they can only replicate at 30 degrees. Other origins need some protein like a PIR or Coli2, and this class over here, the temperature sensitive ones and the protein dependent origins are the ones you really need to pay attention to, because sometimes the strains that we're working with don't have these. So if you're working with a plasmid or you're doing an assembly in a plasmid that has a Coli2 or an R6K or a temperature sensitive origin, you need to make sure that you're going into a strain that has whatever proteins your origin of replication needs, or that you plate at the appropriate temperature. And not only do you have to plate, but you have to keep the bacteria at that temperature or in those conditions the entire time. So when you're growing them up in culture, in liquid culture, so that you can mini prep them, it's the same deal. So that's an easy way to mess up an experiment, by forgetting about the origin. Again, antibiotic resistance, I'm not going to go over that one. That one's pretty obvious. Just pay attention to your antibiotics. And that's all I could really think of for now. So that's kind of a more general look at assembly, how it works. And again, we just did A, B, C, D. You can extend this all the way out to Z if you really wanted to, though I would really pity you if you did have to do that. But shoot me any questions if there's anything that is unclear, and I'll be back soon with more.